Whenever we talk about metabolism, we need to talk about enzymes. And hopefully you remember that enzymes are proteins. And you can see here are the reactants. These are the things that we're putting in. Here are the products. So we're changing our reactants to something else. And you can see this purple line is the amount of energy that you need to put in order to go from reactant to product. Okay, this is that purple line. And so reactants will generally always tend to turn into products, but this reaction may take, 20 years or something like that to actually have the right exact conditions in order for it to take place. It's very, very, very slow most of the time if you're going with just this purple line. But when you add an enzyme into the system, when you add in an enzyme, it's this red line. And so you have to actually put in much, much less energy to go from your reactants to your products. Or let's say to go from glucose to pyruvate or whatever it happens to be. And so enzymes lower the activation energy. They lower the activation energy of a chemical reaction. And basically enzymes really allow life to take place, because without enzymes, really the reactions, the many, many, many reactions that take place in our bodies all the time would take far too long for us to ever be able to live, really. So, and again, enzymes lower the activation energy of a chemical reaction. So, here's just kind of a schematic. Don't worry about all of this. Don't worry about all of this text. What this is showing is this is a generic enzyme here, and this is a substrate. So this would be something like your glucose molecule, whatever it happens to be. This is the thing that you're going to ultimately change. And so the substrate comes in and it does what's called binds to the active site. Basically, the substrate's just going to go and hang out right here, and you can see that it's pretty close to fitting, but then when it actually comes in and binds, the enzyme slightly changes its shape to accommodate the substrate. Not as important. But then, here we have the enzyme product complex, or this is the enzyme substrate complex, and then the enzyme helps to change this into its final form, and it becomes the enzyme product complex, and then the products leave. They exit the enzyme, and the enzyme goes back to this original shape. Okay, so what happened here? The substrate came in, bound to the active site, then the enzyme lowered the energy required to turn this triangle and this semicircle thing into its separate components. Okay, so that's what I want you to know. Okay, enzymes lower activation energy, and they have this active site that substrates need to bind to in order for them to work. This idea is called allosteric inhibition. I should have deleted this text. But basically what happens is there is this thing called an allosteric site. Notice that the allosteric site is not the same as the active site. And what will happen is something like this orange square will come and bind to this allosteric site, and you'll see that the enzyme will actually change shape. And so now the substrate can't fit. The substrate can't get in there because something has bound to the allosteric site. Allosteric inhibition is also commonly referred to as non-competitive inhibition because it's not competing with the active site. It's somewhere else. It's in this allosteric site. We also have allosteric activation, which is basically just the opposite idea. Your activator comes in and binds and then causes this enzyme to change shape so that its active site can interact with this substrate. So there is also the opposite idea of this, which is called competitive inhibition. Competitive inhibition, and what would happen with competitive inhibition is just something that's not the substrate would come and just block the active site. So this is just showing that metabolic pathways most of the time are using multiple enzymes to get from point A to point B. And so here the substrate comes and binds to our enzyme, and then our enzyme changes this this green triangle to a purple square, and then this thing binds to another enzyme, which changes it to another intermediate substrate, and that's going to bind to our last enzyme here, which is going to make our end product. So we went from a green triangle to a yellow square. It's also showcasing here the fact that this can be an inhibitor of our original enzyme. So if this yellow square binds here, the process won't even get started. And this is just a way in which cells can regulate themselves. So if you have a bunch of these yellow squares, you obviously don't need to make more. And so this is going to just inhibit the enzyme that starts the process, that starts converting these green triangles into purple rectangles. This is referred to as feedback inhibition, okay, where the final product inhibits the process from starting, or it could inhibit elsewhere along this path. But this is called feedback inhibition. So we're going to talk in this chapter a lot about energy. Okay, and the cells use the form of ATP, A being this adenine molecule here, and TP meaning triphosphate, 1, 2, 3 phosphate groups. Okay, and this is the basic energy currency of the cell. Cells can use other things, but this is kind of the most commonly used one. Okay, and we're going to get into this process of glycolysis in the next few lectures. I just wanted to show you. Here it is, it's sort of diagrammatically, and this is a glucose getting converted into those 2 pyruvate molecules. Here is that redox reaction of NAD getting converted into NADH, and you're also making a couple of ATPs here. Okay, so definitely more to come about glycolysis later. Just wanted to spend a few minutes talking about general metabolism here.